Welcome back to Intro to Biological Anthropology Lab. I'm David Leitner, I'm your instructor. Today we're going to be tackling a more complex version of the Punnett square called the dihybrid cross. So in a normal Punnett square, you'll remember that we are only sort of looking at a single gene and its alleles, right? And the way we do it, we put the genotype of one parent up here and the genotype of the other parent over here. And then we fill in the boxes, right? A lowercase, lowercase, right? And that's that. That's your Punnett square. It's easy peasy. In essence, a dihybrid cross is no different. We are essentially doing the same thing. There's just one extra little step you have to do that seems a little complicated at first, but I'm going to give you a trick to make it a little bit easier, okay? So, say you have two parents. You've got, say, the female is, in this case, I'm looking at the traits of the pea plants that Mendel was looking at here. So in this case, you have a female whose genotype is heterozygous for yellow and heterozygous for round. All right, and in this case, we made the male the same thing, just to make things a little easier. That's great, but how do we know how to divide that into these cells? Your first thought might be, oh, we just put Y, R, R. You'd be wrong, though, unfortunately. That doesn't help, because what we need is, remember that in Punnett squares, each of these cells represents a potential gamete, okay? This is a potential sex cell, the result of meiosis. So we're looking at the different combinations of this gene and this gene that could end up in those gametes. So to do that, we have to understand that we're looking for a combination of things. So let's consider these two different genes. Now, the way to figure out all the different combinations is simply to use an old trick from algebra for solving, I think it's binomial equations. FOIL, first, outer, inner, last. What does that mean? Well, that means we take the first terms of each pair, okay, like that, and we'll put those right here. So we have homozygous dominant for each, and then we've got outer, which is going to be this and this. So we end up with dominant yellow and recessive wrinkled. And then inner is going to be this and this. So we have recessive yellow, dominant round, and finally last we take this one and this one. All right, and that gives us both of these are recessive. Okay, now we can do the same thing along here because we know this is the same genotype. So okay, so that is the very first step. You have to get each of these individual cells filled, and remember that they represent all the potential combinations of these two alleles in the gametes that are formed in meiosis for these individuals. So then all we have to do is go to the intersections of these boxes, right? And fill them in. So this is going to be, we borrow a Y from over here and a Y from up here. They're both dominant, so boof. We've got homozygous dominant. And again, both of these are going to be dominant, so they're going to be homozygous dominant not only for the color, but for the shape. Okay? And then I'll quickly fill all of these in and come back. Okay, so now that I've filled it in, you can see all the different combinations that the fertilized offspring, fertilized egg would have after combination of each of these individual gametes, right? So these are all the possible combinations, and we'll notice that some of them repeat, okay? So while we only have one that is dominant, homozygous dominant for both genes, we've got two that are homozygous dominant for color and heterozygous for shape. 1, 2, see, like that. And we can go through and count all of these here, and we end up with this sort of frequency distribution up here. Looks very complicated. But that's not the only thing we can count. We can also go through and count the distribution of phenotypes. So these are all the possible genotypes. Now we can go through and calculate all the phenotypes. And the way we do that is very simply just looking at each pair and asking ourselves, what trait would this express? So I'll start with one, and then I'll fill them all in and let you, and we'll jump ahead here. So, for instance, homozygous dominant is obviously dominant. That's an easy one. Homozygous dominant and heterozygous, well, we just go with the dominant one again here. And I'll go ahead and complete this and come right back. Okay, just to help us visualize this a little better, I've gone ahead and colored everything in, and that makes it a little easier to count. And we'll notice that we've got a whole lot of these that are yellow and round here, right? In fact, actually, if we count them up, we get 9. Then if we count up the others, we have 3 that are yellow wrinkled, and 3 that are green round, and only 1 that is green wrinkled. Okay, why is that? Well, remember that dominant traits are going to be more plentiful just because they will express themselves in heterozygous individuals. So you don't have to be homozygous dominant in order for the dominant trait to show up. It'll show up in these heterozygous pairs as well. So that gives you a lot more. 1, 2, 3, 4, 5, 6, 7, 8, 9 to be exact. Okay? Whereas in order to have the recessive trait, you can't have any dominant alleles whatsoever. So it's going to be less likely. It's going to be less abundant in this distribution. And in fact, actually, there's only one cell where there are no dominant traits at all out of all of these. 1 out of 16 combinations that is homozygous recessive for both traits. But I want to demonstrate something else here. You'll notice that if we just take one trait at a time, we'll notice that all of the recessive color traits, that is the green, add up to 4. There are 4 greens in total, and you can see that here. These are the 2 possibilities with green. That adds up to 4. 4 out of 16 is 1/4, which is 25%, which is the same frequency we would expect for homozygous recessive in our ordinary Punnett squares that we've done, right? So I'm not going to fill in the whole thing here, but 1 out of 4 will be recessive, okay? Or will exhibit the recessive trait. So anytime, now not every combination is going to end up with these distributions. This is just what you get when you combine 2 individuals that are heterozygous for both traits. So as you can see here, heterozygous, 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 heterozygous. And you'll notice, again, their phenotypes are the same as the most abundant one because the recessive traits aren't activated. So I think that about caps it out. The main thing I want you to take home from this is that this is no different from the Punnett squares we did last week. It's just this added step of trying to figure out all the combinations of the possible gametes that we have that trips people up usually. Okay, one way to keep from not getting confused is always remember in any pair you're going to list in any pair of alleles you're always going to list the dominant one first. That helps a little bit regardless of whether you're reading the column or row. And always keep the same order of genes as you're writing things down. So you notice I put the color gene ahead of the shape gene in every one of these because that's how it was written in the genotype of the parents. That is just that being consistent that way helps keep you on track and helps you from getting confused or making little mistakes here and there that add up. Okay, so I hope that helps. Have a great week, everyone, and I will see you soon.